Dengue is a mosquito-borne viral disease transmitted by the bite of an Aedes mosquito in humans. According to World Health Organization, dengue is considered as one of the fastest emerging infections in the world. The incidence of dengue has increased 30-fold over the last 50 years. Up to 50 to 100 million infections are now estimated to occur annually in over 100 endemic countries, putting almost half of the world's population at risk. Dengue cases are noted to increase due to growing urban population and number of mosquito breeding sites such as open water containers like drums, bottles, tin cans, jars, roof gutters, old tires, and flower vases. The Philippines is considered as one of the countries that are at risk of dengue transmission. In December 2011, the Department of Health recorded 118,868 dengue cases, with 639 deaths nationwide. Majority of these cases are children aged 1 to 9. The DOST OL trap. To address the problem of dengue, the Department of Science and Technology Secretary Mario G. Montejo made a commitment to create a technology through the Philippine Innovation Projects of DOST. This encourages complementation, where initiatives are harmonized for the various disciplines to work together with a goal of making things simple yet beneficial. The Industrial Technology and Development Institute, in cooperation with the Philippine Council for Health Research and Development, developed a technology known as the Ovicidal Larvicidal Trap System, an intervention to reduce dengue cases in the country. The DOST-OL trap system consists of black plastic cup, lawanite, and organic pellet. It is a simple technology that lures and catches Aedes aegypti mosquitoes and kills its eggs and larvae in the process.
DOST Natural Larvicide for Dengue Vector Control Program. The ITDI DOST conducts 3 phases involved in this program. Phase 1 is the laboratory study on the development, performance testing, and fabrication of an ovicidal trap system for Aedes mosquito. Phase 2 is the field testing in Quezon City and Marikina, done on September 24, 2010. And Phase 3 is the rollout of DOST-developed ovicidal larvicidal trap system nationwide. In a joint collaboration between 2 national government agencies, the Department of Science and Technology and the Department of Health, with a common goal to reduce dengue cases in the Philippines, an initial 200,000 OL traps were distributed for 50,000 households nationwide. To formalize the partnership, a Memorandum of Understanding was signed last April 14, 2011, that resulted to the distribution of additional 500,000 OL trap kits nationwide. In the Calabarzon region, Dr. Alexander R. Madrigal spearheaded the regional turnover of 11,200 OL trap kits with a total of 2,800 household beneficiaries. 5 cities and municipality, namely Lipa City, San Pablo City, Santa Rosa City, Calamba City, and Ibaan, Batangas, were identified as the first beneficiaries of the OL trap system in the region. Additional municipalities and cities were also included as beneficiaries to receive the free OL trap kits. These are Pagsanhan, Los Baños, Calamba, and Siniluan in Laguna, Mauban in Quezon, and Antipolo City in Rizal. Through an interagency collaboration between the Department of Health, Department of Science and Technology, Department of Education, Department of Interior and Local Government, Department of Environment and Natural Resources, and Metro Manila Development Authority, the efforts to control dengue in the country are sustained. A memorandum of agreement known as Aksyon Barangay Contra Dengue was signed on August 25, 2011 in Barangay Bagbag, Quezon City. This becomes a manifestation of a strong political commitment and resources for improved and sustained efforts towards a successful prevention and control of dengue cases in the Philippines. To further strengthen the government's campaign to reduce the number of mosquito population, thereby reducing transmission of the virus, DOST spearheaded a program of deploying the OL trap kits to all public schools nationwide in collaboration with the Department of Education, Department of Health, and Department of Interior and Local Government.
Role of participating agencies. DOST will conduct an eco-orientation training for the provincial science and technology directors and DepEd representatives in the regions. Deliver the OL traps to DepEd division offices. And consolidate monitoring reports from DepEd division or regional offices. DepEd will assign a coordinator for the region, the division, and the school to oversee and supervise the project implementation. Transfer OL traps from the division office to the recipient schools. Conduct orientation training in each recipient school before project implementation. Organize and utilize the school dengue brigade in the project implementation, specifically in other intervention activities. And ensure proper installation and servicing of OL traps. DOH will designate a coordinator per region. Assist in the conduct of eco-orientation training for DepEd representatives in the region. And consolidate the dengue incidence data in the community. DILG will provide data on dengue cases to partner agencies and extend assistance during the conduct of orientation training. Process flow of the OL trap school-based rollout. The issuance of memorandum circular for the project implementation by the DOST and DepEd. The conduct of orientation training in the provinces by the DOST, DepEd, DOH, and DILG regional offices. The training will be attended by the DepEd district or division officers and 2 representatives per school. Conduct of reorientation training per school 1 week after the provincial-wide orientation training. Installation of OL traps on Thursday, a week after the reorientation training, and servicing of OL traps every Tuesday. That will run for 6 months. And lastly, monitoring of OL index in the school and dengue incidents in the community for 6 months by DOST, DepEd, and DOH-4A. A vector is an arthropod that is capable of acquiring, supporting the development, and transmitting a pathogenic agent such as bacteria, virus, and parasite from one host to another. Dengue is transmitted by a vector known as Aedes mosquito, particularly Aedes aegypti and Aedes albopictus. The biology of Aedes mosquito. The Aedes aegypti is the primary vector carrier. It is involved in cases of epidemics in urbanized areas. It is confined in artificial containers and rests in cool, dark corners of the house. The Aedes albopictus, on the other hand, is the secondary vector carrier. It maintains the virus in the environment, especially in the rural areas. It prefers to breed outdoors in natural containers, like clearings and vegetations. The morphology of Aedes mosquito. In terms of morphology, both species is 3 to 4 millimeters in size and dark brown to black in color. The thorax of the Aedes aegypti is lyre-shaped with white marks or scales, while Aedes albopictus has single white marks. The abdomen of the Aedes aegypti has white scales, while the Aedes albopictus has white and dark marks.